Hello everyone, my name is Ronak Ramesh, and I'm a senior at the Academy of Aerospace and Engineering this year. And today, I'm excited to present my research on comparing and analyzing point mutations through molecular dynamic simulations to test protein structural stability and simulation validity. So without further ado, let's begin. So here's a little overview of what I'll be talking about today. First, I'll start off with an introduction, and then I'll talk about some background information on my topic, and then I'll delve into my research objectives and what my research aims to do. Then I'll talk about methods and results, and then delve into my conclusions and discussion of my data. And then finally, I'll touch upon my future steps and acknowledgements. So, have you ever wondered how tiny changes in our DNA can lead to life-threatening diseases? Because that's the essential question I'm hoping to solve with my research. As you can see on the screen, mutations in protein coding regions account for approximately 70 to 80% of disease-related mutations in today's society. This includes diseases like cancer, cystic fibrosis, sickle cell anemia, and so much more. This is a serious problem as many human lives are in danger as we speak. And the scary thing is, even the smallest of mutations and changes in our DNA can lead to these types of catastrophic diseases. I've included this diagram here on our right to illustrate just how these single amino acid mutations, otherwise known as point mutations, really work. And as you can see, even a single amino acid residue switch can change the overall protein structure sequence, which completely changes how the protein and how it works in our human body. So understanding the impact of these point mutations on protein structure is extremely important in today's society, as it can help us understand these diseases like cancer and cystic fibrosis better, and therefore it can help us develop cures and treatment plans as well as prevention plans. However, that's easier said than done. Predicting the impact of these mutations is quite tricky, and current and traditional plans and experimental methods for studying these protein mutations, which involves studying proteins in like a lab-based setting, for example, can also be time-consuming, expensive, and often impractical. And that's where my research comes in. So my research aims to investigate the structural changes induced by point mutations in human proteins and assess their impact on stability and function. Additionally, the study seeks to validate molecular dynamic simulations as a computational tool for studying protein mutations in the human body and explore the potential of computational techniques for informing therapeutic interventions. So these computational methods provide a cost-effective and efficient means of analyzing the structural changes induced by mutations. And by combining experimental data and computational simulations, I hope to gain valuable insights into disease mechanisms and hopefully pave the way for the making of personalized and targeted patient therapies. So this research is very important because, as I mentioned before, protein mutations play a crucial role in the progression of various human diseases, and understanding how these mutations affect protein structure and function is essential for developing targeted therapies and improving healthcare outcomes for patients with these types of genetic disorders. So I've talked a lot about molecular dynamics. So what even are molecular dynamics? So while we cannot directly see how proteins move and interact inside cells, molecular dynamic simulations, or MD simulations, offer a powerful tool to visualize and understand their dynamic behavior. So by calculating the forces between atoms based on their positions and properties, MD simulations simulate the protein's motion over time and create a movie-like representation of its journey, like the video I just played on the left here. While the underlying calculations are quite complex, just understanding the basic principles of MD can open the doors to exciting discoveries in the world of proteins and mutations. So for my methods, I started off with my research by using the Humzavar dataset, which contains over 70,000 single amino acid substitutions in human proteins found in clinical research. Now, a way as scientists study proteins is by growing crystals of them and then using X-rays to determine the precise arrangement of atoms within the crystal. And these structures are then stored in databases like the PDB database, otherwise known as the Protein Data Bank. So to obtain the crystal structures of the proteins for my research, I used this exact database online, and I selected proteins relevant to my objectives from this database. And then from those structures, I then incorporated them into MD simulations. And by using Yale University's high performance clusters called Grace and McLeary, I was able to extract efficient data analysis. And then I was also able to run RMSD and RMSF calculations, which I'll get into further later. So for MD simulations, these are the steps that I use to conduct them. So first I start off with topology generation. And in this step, the topology of the system is generated, which includes defining the types of atoms, bonds, angles, and dihedrals which are present in the molecule. And second, the box definition and solvation. So this is where the simulation box is defined, and this is where we place the molecule of interest. And so solvent molecules, usually water, as you can see here, surrounds the molecule to simulate and mimic the protein environment. And so third step is ion addition, and this is where ions are added to the system to neutralize any charge present and basically make sure that the system is electrically neutral. The fourth step is energy minimization and equilibration. So the initial system goes through an energy minimization to achieve a stable starting structure. And then the system is then equilibrated to reach like a steady state before production runs. And so I like to think about this process kind of like smoothing out wrinkles in a bed sheet before making the bed. So we let the system relax until it's nice and calm. So the fifth step is the main MD simulation. And this is where trajectories are generated, capturing the movement of the atoms. And so we watch how our molecule moves over time, like a movie, like we saw before. And afterward, we study the movie to see what's really happening inside our molecule. So RMSD, also known as root mean square deviation, measures the average distance between corresponding atoms in two protein structures. So in my graph here, I made three key comparisons to evaluate the structural changes induced by mutations and protein dynamics. So here you can see XTIL, and that's just short for crystal. And so these crystal structures served as the starting points for my MD simulations, as they allowed me to compare the behavior of the proteins in their crystal states and the simulated MD states. So in this first blue bar here, that shows the comparison between the crystal wild type and the mutated crystal protein structure obtained from the PDB database, and it just showed me how much the mutation has moved the backbone atoms from their original positions. And so the RMSD calculated value for this was approximately 2 angstroms, which shows me kind of a moderate degree of structural deviation from the mutation. The second green bar here is a comparison that assessed the fluctuation of wild-type protein structure obtained from molecular dynamic simulations around the crystal wild-type structure, and it kind of gave me insights into the inherent flexibility and dynamic behavior of the wild-type protein in a simulated environment. And so the calculated RMSD value for this comparison was only 1.7 angstroms, which showed me a close correspondence between the molecular dynamic simulations and the experimental structures. Now, the final bar here, the yellow bar, is the final comparison, which evaluates the fluctuation of the mutated proteins from the MD simulations around the crystal mutant structure. And so it just quantifies just how much mutated structure fluctuates around its average position during the simulation. So the calculated RMSD value for this was approximately 4.3 angstroms, which showed me a higher degree of structural deviation. So another data analysis tool that I used was RMSF, which is the root mean square fluctuation. And so this just indicates the flexibility of protein atoms over the course of the simulation. So high fluctuations suggest increased flexibility, and it shows me regions of structural instability and potential therapeutic target regions. So notably here at the mutation site residue 21, there's a significant peak in fluctuation, which means like heightened flexibility in this region. And that was to be expected because this was where the mutation site occurred. And it's expected that there's more flexibility and fluctuation at the mutation site because when a mutation occurs at a specific amino acid residue within the protein structure, it can disrupt the normal interactions and stability of the surrounding residues as well. So just like a ripple in water causes nearby areas to move, a mutation in a protein can cause nearby amino acids to become more flexible, and it leads to increased fluctuation or movement within the protein.
You have just about 2 minutes remaining.
Sounds good. Thank you.
Yep.
So for conclusions, I kind of came to the conclusion that by cross-referencing simulation data with crystal structures from the PDB database, I was able to validate the accuracy of molecular dynamic simulations. So for example, when I found that the calculated RMSD value of the comparison between the crystal wild type and the MD wild type was only 1.7 angstroms, it showed me just how accurate the MD simulations really are. Not to mention, MD simulations are also very cost and environmentally friendly as they are conducted like completely through a virtual environment. And by using these types of computational resources like MD simulations, we can conduct research more efficiently while also minimizing resource consumption and waste generation. So other insights from this research can have significant implications for therapeutic interventions. So, by understanding how mutations affect protein structure and function, specifically how they become exceedingly unstable at the mutation site, as shown by the RMSF graph, we can start to make targeted therapies to restore normal protein activity and improve patient outcomes. I like to also end on the note that um, at the end of the day, I didn't just pick up this research project or work really hard for this research just to compete at a science fair. I really want to make an impact in healthcare, and I hope that this research can just be the first steps of that process. And then for future steps, there are several different ways I want to take this research. So firstly, I want to expand my analysis to include a wider range of protein mutations and explore their effects on protein function more in detail. And I also want to incorporate more advanced computational techniques and hopefully machine learning algorithms to enhance the predictive power of simulations and provide deeper insights into disease mechanisms. And
Great.
ultimately, my main goal is to translate this research into practical applications by making targeted therapies for specific genetic disorders and contributing to advancements in personalized medicine. And just for my acknowledgments, I'd like to thank my school um, and my lab at Yale for supporting me throughout this research process. I would also like to thank AEOP, the Army Educational Outreach Program, as it was through them that I met my research group at Yale and discovered this fascinating world of biology. And I would also like to thank all of you for listening to my research, and now I'm completely open to any questions you might have. Thank you so much.